NADH in the cytoplasm to the NAD plus in the matrix. Right? That's the purpose of the electron shuttle, because the cytoplasmic NADH itself does not cross the inner membrane of mitochondria. We have a point when we talk about cytoplasmic NADH produced during glycolysis. First, it has to come through the outer membrane of mitochondria. Right? So the outer membrane of mitochondria has the pollen. But how pollens are established, right, on the outer membrane of mitochondria briefly. So the NADH produced during glycolysis in the cytoplasm first transported into the inner membrane space. From there, the electrons will be shuttled inside the matrix. Therefore, sometimes we ignore the outer membrane of mitochondria, so just call it cytoplasmic NADH. I think that's what you're talking about. Right? The cytoplasmic NADH produced by glycolysis or during glycolysis. This NADH is not transported across the inner membrane of mitochondria because the NADH cannot come across the inner membrane of mitochondria. So the electron has to be shuttled across the inner membrane of mitochondria by the malate aspartate shuttle. Yeah.
Well, I was just going to ask something about, on your slide it says they come in through boron channels too, so that's how some of them get into the, because in your slide you have the NADH swapping, doing the shuttle thing on both sides. So
yeah,
I can get into that, right?
Two types of NADH. One in the cytoplasm, the other one in the matrix. So there are two different classes.
And they never leave, they stay in the
Yes, the one inside the matrix does not come out of the matrix of mitochondria.
There's just a continually increasing amount of NADH in the matrix, and then increasing.
Oh, let's just put it that way, because there are two groups of NADH. At the molecular level, they're the exact same. But one is in the cytoplasm, the one in the matrix. They do not exchange themselves.
Wait,
Yes.
so do the electrons that come in, are they used to make the NADH inside the matrix?
So the electrons are shuttled from the cytoplasmic NADH to the matrix NADH. Right? That keeps it simple. So the NADH itself does not come across the inner membrane of mitochondria. The electron harvested during glycolysis has to be transported across the inner membrane of mitochondria by the shuttle.
As this continues though, wouldn't there be an increasing amount of NAD plus in the cytosol and an increasing amount of NADH in the matrix?
Yeah, it goes through glycolysis.
Right? Exactly. So, in order for glycolysis to occur, you need a NAD plus. Right? If you don't have NAD plus, you're going to inhibit the glycolysis. That exactly happens when, in the lack of oxygen, what happens? In yeast, we call it fermentation. In mammals, in our cell, animal cells, it produces lactic acid while you produce NAD+. Therefore, glycolysis can continue. Now, the NADH inside the matrix of mitochondria, in the matrix of mitochondria, what does it do? It contributes, it donates electrons to the electron transport chain. Any questions related to it? These are good ones. If I didn't make it clear before, the two groups of NADH, okay, they do not exchange. Yes, sir.
How does the NAD plus get in the inner matrix in the first place?
That's a good question. I'm not going to test you. I asked that question before. What is NADH? What does NAD stand for? Nicotinamide dinucleotide phosphate, right? So it can be synthesized in the mitochondrial matrix, right? When it's synthesized, it does not leave mitochondrial matrix. Similarly, it can be synthesized in the cytoplasm. Yeah, Trey?
No, I understand it better now.
Okay.
I just needed to like know the difference as like.
Yeah, the
just
purpose
like-
of the electron shuttle is because the inner membrane is tightly composed. We talk about the phospholipid bilayer from the layers, right? And the outer membrane of mitochondria is more porous, or we call it semi-permeable. The inner membrane is more compacted, therefore it can establish the proton motive force, the electrochemical gradient of proton. Any questions?
Do we need to know the specific signal for when a vesicle becomes uncoated?
So what's the signal drives vesicle? What signal drives the disassembly of the coat? Right? What signal drives the disassembly of the coat? It's the first question today. You were there. The first eye-click question we had today. Hydrolysis of GTP, right, leads to the disassembly of the coat. So the sequential event is a G protein, right, recruits the coat to the donor membranes to form the vesicle, and typically the coat acts as a gap. Therefore, the G protein will hydrolyze the GTP into GDP and PI. And when the G protein is associated with GDP form, then leads to the disassembly of the coat. So this is both the case, all the case for COP2, COP1, and Claflin coated vesicle. But by now you know the G protein for COP2 and the G protein correspond to COP1 and Claflin vesicle. Yeah.
What was the, was there any importance relevant to something we learned before or afterward for the non-Mendelian segregation of mitochondrial genetics?
Okay, so the question is,
Was that related to the electron transport chain or photosynthesis as well? Or is that?
not related to the electron transport chain. So, Amber's question is asking, I discussed with you for mitochondria inheritance, particularly in, let's forget the yeast, okay? In animals, in mammals, we maternally inherit mitochondria. In animals, well, not most, almost exclusively, because the sperm does not contribute to mitochondria, so inherited by inherited maternally, right? So the example I used in the lecture is for yeast mitochondria during cell division. These mitochondria, now we have two species. One is antibody resistant, the other is not. These two species, when they mix in the diploid, and then when the diploid divides, its progeny can inherit either one type or the other, not both. So that suggests the mitochondria genome is not strictly transmitted into the progenies like the nuclear genome. Nuclear genome, we know, is strictly Mendelian inheritance, right? Always Mendelian inheritance. When the cell divides into two daughter cells, the genetic information is equally participate. And then go into meiosis, we have exchange of genetic information. Always stick to the Mendelian segregation pattern, but mitochondria genome does not. That's the point. But this has nothing to do with the electron transport chain. That suggests that, for example, during cell division, mitochondria is not strictly segregated, like the chromosome we will discuss later on. Strictly segregated, for example, cis chromosomes separate from each other during mitosis. Okay, mitochondria does not.
Okay, I have a question about, mitochondrial import of proteins. So I know today you said that HSC70 was the chaperone that prevents the
Folding?
Yeah, the folding. So when the N-terminus goes in the import pore, and then once it's going through the two pores, you have HSC70 again
In the matrix.
in the matrix. Why do you need it in the matrix if it's gonna fold once the N-terminus is pushed off?
Yeah, so the question here is, what are the function of the chaperones, in this particular case, HSC70, in the cytoplasm and in the matrix? So in the cytoplasm, we discussed that the function is use ATP hydrolysis to prevent folding. So similarly, in the matrix, the chaperone is required by ATP hydrolysis. The idea is it's required for pulling the peptide through the protein channel. Okay. Now, it may also involve counterintuitively. You have a good question that in the matrix, this chaperone facilitate folding. So you can ask this question: why in the cytoplasm this particular chaperone prevents folding? Why in the matrix this chaperone is required or promotes folding? Okay. For simplicity, these chaperones themselves are regulated by others. All right. So let's just take it at face value. In the cytoplasm, this chaperone prevents folding of the peptide synthesized by the ribosome. Why, when the peptide gets into the matrix, now this chaperone is regulated by the others. So it promotes folding into the final conformation. In fact, I didn't talk about the slide. One of the slides says another chaperone called HSC60 is involved in folding of the peptide in the matrix.
And that's what helps the HSC70 promote.
Yeah.
You're talking today about nuclear transport. You mentioned there are three main parts on the nucleus.
Nuclear pore complex.
Yeah, on the nuclear part complex. First for the scaffold.
Yes, it was a scaffold.
Then the protein that forms the nuclear basket. What exactly does the nuclear basket do? Does it just collect the proteins that come in and take them off your own?
Okay, so the question is, you guys probably realize if you went to today's lecture, because I tried to include the nuclear pore complex in our discussion, will appear on the test, so I sort of rush. Do we already have a pen? Why? Looks like someone is sitting on a pen. I think it's my office. Get one. You got one too? Thank you. For the nuclear pore complex, in general, we have three components. One is the scaffold part. Subcomplex, you can think about scaffold subcomplex. The other one is involve the membrane anchor complex, subcomplex, because it's embedded into the nuclear envelope. And the other one you may realize from the basket is called the FG filament subcomplex. This is basically is the basket of the nuclear pore complex, where controls the size of the protein that can be freely permeable to the nuclear pore complex. So less than 40 kilodalton we talk about right today. Molecules that smaller than 40 kilodalton can diffuse through the nuclear pore complex. So the scaffold provides the structural support of the nuclear pore complex, and this is the membrane anchor. Helps the nuclear pore complex establish embedded